Good afternoon, everyone. Welcome. I'm Jack Van Horn from the University of Virginia. It's my distinct pleasure to welcome you to this installment of our 2025-2026 Foundations of Biomedical Data Science seminar series. Our unique lecture series is made possible through the generous support of the University of Virginia's College of Arts and Sciences, the UVA School of Data Science, and through a grant from the National Institutes of General Medical Sciences. Since its founding in 2016, the Foundations of Biomedical Data Science Seminar Series has been at the forefront of exploring the transformative impact of data science methodology, artificial intelligence, deep learning, and statistical modeling across diverse themes in biomedical and health sciences. Our selected theme for this year is focused on data science approaches and the study of autism spectrum disorders, where every Friday, we enjoy a range of talks from leading thinkers about the issues, the promise, opportunities, and hurdles associated with research involving autism spectrum disorder. This includes deep phenotyping, clinical assessment, advanced genomic, transcriptomic, neuroimaging, metabolomic, and other methodological approaches, along with a wide variety of data analytic opportunities presented for data science, deep learning, artificial intelligence, and spatial modeling. We are particularly excited for this year's seminar series program, where biomedical research and computational experts will shape for us the domain of autism and provide insights into its quantitative analysis. This topic is very newsworthy right now and subject to much debate, possible misunderstanding concerning its issues, its causes, and its treatments. Clearly, bringing data and novel methods to bear to understand the roles of genetics, the developing brain, and the environment will be very important to explore in autism. So we're very excited and looking forward to covering the breadth and depth of this novel topic area of research and investigation. For today's seminar series, I am delighted to welcome our speaker, Professor Katherine Lawrence from the University of Southern California. Katherine is an assistant professor at the USC's Mark and Mary Stevens Neuroimaging and Informatics Institute, and she is a co-chair of the Enigma Autism Working Group, an international consortium advancing large-scale reproducible neuroimaging research in autism spectrum disorders. Her work leverages large-scale neuroimaging data sets and analytic methods to improve our neural understanding of the neurodevelopmental conditions of typical development, including their immense individual variability and applications in conditions like autism spectrum disorder. She is a recipient of an NIMH K01 award, and her research has been funded for over a decade by organizations including the NIH, the Thrasher Research Fund, the ARCS Foundation, and the Momental Foundation, and has earned recognition from the International Society for Autism Research, Society for Neuroscience, and other various entities. In the spirit of our 2025-2026 Biomedical Data Science Seminar Series, today's lecture is entitled Leveraging Large-Scale Neuroimaging to Dissect Heterogeneity in the Neural Mechanisms of Autism. In her talk, Dr. Lawrence will present work leveraging large-scale resting state fMRI, task-based fMRI, and diffusion-weighted MRI datasets to rigorously dissect how individual differences in age, sex, and genetics shape brain connectivity and activity in autism. She will also present her work using advanced microstructural models to robustly identify normative sex differences in white matter microstructure across the lifespan, providing an important foundation for understanding sex differences in ASD. Together, this work helps to map how individual differences impact the neural heterogeneity observed in autism and underscores the importance of characterizing individual variability to best support unique strengths and needs of every individual. As we do, we are streaming this lecture live and for recording via YouTube. So if you're watching on YouTube, thank you so much for joining us. Also, our specially selected 2025-2026 Biomedical Data Science Innovation Lab participants, as well as alumni, are encouraged to submit any questions they have via the chat feature in their YouTube sessions. I will be synthesizing these questions and I will ask them on your behalf during the last 10 minutes or so of the lecture. And with that said, thank you so much, Katherine. We're really looking forward to having you tell us about your work.
Thank you so much for having me. I'm really excited today to be presenting my work using large-scale neuroimaging to dissect heterogeneity in the neural mechanisms of autism. I'm going to start by presenting our research on the neural mechanism of autism, and I'll be using flexible language throughout this presentation, which is to say that I'll be using both identity first and person first language when talking about autism. I'll then move on to presenting our work, looking at brain-based sex differences across the population to help lay a foundation for better understanding sex differences in autism and other conditions that commonly co-occur with autism. So, starting with our work on autism, I'm going to present our work using resting state fMRI and task-based fMRI to investigate intrinsic functional connectivity and brain activity. And I'm then going to present our work using diffusion-weighted MRI to look at white matter microstructure. So let's go ahead and get started. Autism is very heterogeneous, and this heterogeneity is a really big challenge for developing broadly effective supports for autistic people who want such supports. And neuroimaging is a very promising way to parse this heterogeneity. For this reason, we're very interested in using neuroimaging to dissect brain-based variability in autism with a long-term goal of informing how we can best support the diverse profiles of strengths and needs within the autistic community. For this series of studies, we started by focusing on 3 core sources of heterogeneity in autism. First, we focused on sex effects in autism. Autism exhibits substantial sex differences in prevalence. It's 3 to 4 times more common in boys than girls. And at the time we did these studies, almost all imaging work in autism had focused on samples that were mostly male, meaning we really had no idea if our understanding of autism would generalize to girls with autism. Second, we examined age effects in autism. Previous studies completed separately in children and in adults had yielded conflicting results about how functional connectivity is altered in autism. This suggested that if we want to better personalize treatments or create biomarkers for autism, age may be a crucial variable to consider, but no work had examined the longitudinal age trajectories. Third, we focused on the impact of genetic load for autism. Autism is highly heritable, with a heritability of 80 to 90%. And the majority of genetic load for autism lies in common genetic variation across the genome. However, no work had investigated how genetic load for autism across the genome was associated with brain function. So as a whole, our goal is to parse the heterogeneity associated with autism by examining how these important sources of individual variability impact brain function. So, starting with sex differences, I'm going to provide a bit more background on sex differences in autism. Not only is autism substantially more common in boys than girls, there's also emerging evidence for different behavioral presentations of autism in boys and girls. This includes girls with autism showing fewer social differences in some respects, for instance, having closer friendships, in addition to displaying different executive function challenges, and showing attention differences, such that social stimuli may be more salient. However, it was completely unknown how the neural networks underlying these symptom patterns may be altered in girls with autism. We therefore led the first ever autism study to examine sex differences in these resting state networks. We leveraged resting state fMRI data collected as part of an NIH-funded multi-site network. And we looked at the within and between network connectivity of multiple higher order networks. The first of these is a default mode network, which is associated with social cognition. The second of these is a central executive network, which is related to executive function and decision making. And the last of these is the salience network, which, as its name implies, is thought to play a role in detecting salient stimuli and coordinating a neural and behavioral response to them. These 3 networks are also interrelated in that the default mode and central executive networks are anti-correlated in neurotypical adults, and the salience network plays a role in switching between these 2 networks. Here we use 2 complementary approaches to rigorously map connectivity differences. First, we use a region of interest, or ROI-based network approach, which is good for looking specifically at the main nodes of each network while still retaining a good amount of power. However, it doesn't tell you how these networks may differ in areas which aren't the main core hubs, which is where the whole brain approach comes in, since this lets us look at network connectivity in a more regionally specific manner across the entire brain. So I'm going to start with results from the ROI-based approach. Overall, we found that autism exhibited altered sex differences, and I'm going to show you how we reached that conclusion. First, we looked at sex differences in typically developing controls, where the stars indicate the significant sex differences. And I've highlighted those sex differences here. For the sake of time, I'm not going to go through each individual difference, but broadly, I want you to notice that there are typical sex differences in salience network connectivity, where the cross highlighted by these gray dashed boxes is showing salience network connections. And you can see that all the stars land within those gray boxes. If we look at sex differences in the autism group, we see none of the typical sex differences in salience network connectivity. Instead, we see the reverse. The sex differences that we do see in the autism group aren't observed in the typically developing group, supporting altered sex differences in autism. When completing whole brain analyses, we again found altered sex differences, such that only the typically developing group exhibited sex differences in salience connectivity, here with the posterior parietal cortex and the central executive network. This pattern of altered sex differences in turn suggests the presence of subtly altered sexual differentiation and functional connectivity in autism on average. This finding is in line with previous behavioral and hormonal work and indicates that biological factors which relate to sexual differentiation, such as sex differential gene expression or sex hormones, may contribute to the presentation of autism and warrant further investigation. I'm now going to move on to our autism study, which looked at sex differences in brain activity to social rewards. So, why do we care about social reward processing in autism? One of the most influential theories of autism suggests that the social differences observed in autism arise from reduced brain activity to socially rewarding stimuli, like a smiling face, which means that infants who go on to develop autism don't pay as much attention to social information, which means in turn that there are fewer opportunities for social learning, which is hypothesized to ultimately contribute to the social differences you see in autism. However, it's important to note that many autistic people find that the social motivation theory doesn't square with their lived experiences, suggesting it may not be relevant for all individuals. Additionally, the studies I've tried to test this theory have only looked at male samples, so we really don't know if this theory is applicable to autistic girls or broadly applicable to autism as a whole. To address this question, we therefore completed the first ever analyses to characterize social reward processing in girls with autism. Specifically, we examined their task-based fMRI data while they were completing an implicit learning task, where the reward that participants received in correct trials was a smiling face. We found that girls with autism exhibited significantly greater activity to social rewards than boys in a key reward region of the brain, the nucleus accumbens. And this was true both when using an ROI-based approach and when using a whole brain approach. When comparing autistic and typically developing girls, we didn't find any difference in nucleus accumbens activity, but we did find that girls with autism exhibited greater activity in other regions of the brain, which I'll go through now. One of these regions is the anterior insula, which is thought to play a role in salience perception and is a hub of the salience network. Autistic girls also displayed more activity in the lateral orbitofrontal cortex and the ventrolateral prefrontal cortex. These areas are known to play a role in reward learning and also encoding reward-related properties more generally. So as a whole, this suggests that girls with autism may find socially rewarding stimuli more salient and be able to process and learn from them better. This has important implications for the social motivation theory of autism by revealing that this theory may not be as relevant for all individuals with autism. I'm now going to move on from talking about sex effects in autism to age effects in autism. Previous functional connectivity analyses on autism have shown that studies focused on children report hyperconnectivity, whereas studies focused on adulthood find hypoconnectivity, suggesting altered trajectories in adolescence. However, because these studies were cross-sectional, we don't know if the observed differences were actually just a function of ascertainment bias. If there are altered trajectories, this is really important to know so that we can appropriately tailor biomarkers and treatments. We therefore completed the first longitudinal analyses ever of functional connectivity trajectories in autism. For this study, we focused on a longitudinal sample from an NIH-funded project that was scanned once during early mid-adolescence and then again during late adolescence. Here we focused on within and between network resting state connectivity of the default mode network, salience network, and central executive network. As previous studies examining each of these networks separately found significant differences in autism. I'm going to start by talking about how the autism and typically developing groups differed within each time point. There were no significant differences at the early mid-adolescent time point, but there were in late adolescence in terms of connectivity between the central executive network and the medial prefrontal cortex hub of the default mode network. And in this graph to the right, I've visualized the whole brain result so you can see the direction of findings. As you can see, at this late adolescent time point, the groups differed such that the central executive network and default mode network were anti-correlated in the neurotypical group, but not in the autism group. We then looked at developmental trajectories separately in the autism and typically developing groups. The autism group exhibited no significant longitudinal changes in functional connectivity. There were, however, significant developmental changes in the typically developing group that are in line with known patterns of connectivity in neurotypical adults. The default mode network showed a significant reduction in short-range connectivity among typically developing youth. Typically developing youth also displayed reductions in between-network connectivity of the salience and central executive networks, reflecting greater specialization of these networks. The main takeaway from this, though, is that the typically developing group showed the expected developmental changes, whereas the autism group did not show any significant changes. When directly contrasting developmental trajectories between the autism and typically developing groups, autistic youth exhibited significantly different trajectories that prevented them from attaining typical adult-like patterns of functional connectivity between the central executive and default mode networks. As the amount of segregation between the two networks has previously been related to executive function, this suggests the possibility that these altered trajectories could be related to the persistent executive function challenges commonly observed in autism. As a whole, these findings critically emphasize the importance of considering age when trying to understand the neural mechanisms of autism. I'm now going to move on from talking about age effects in autism to talking about the association between genetic load for autism and brain function. The bulk of genetic load for autism lies in common genetic variation that is spread across the genome. Any individual genetic variant has a very small effect, but the additive effect of these common variants contributes substantially to autism. However, no one had examined how this cumulative genetic load for autism is associated with brain function, with previous work only examining one or a couple of common genetic variants at a time. Another notable property of genetic load for autism is that females require a significantly greater genetic load to develop autism than males. This effect is thought to be why autism is more common in boys and girls, but no one had directly investigated what in vivo mechanisms may underlie this effect. We therefore investigated for the first time how additive genetic load across the genome impacts functional connectivity in boys and girls with and without autism, with a hypothesis that genetic load would differentially modulate brain connectivity in females and males. For this study, we used a multi-site sample of youth with and without autism. We specifically focused on whole brain resting state connectivity of the salience network, because although other networks, like the default mode network and central executive network, are related to symptoms we commonly see in autism, the salience network is specifically hypothesized to relate to the development of core autism traits, with previous work in infants supporting this. We related connectivity of the salience network to cumulative genetic load, which we quantified using polygenic scores. And we quantified polygenic scores in both our autistic participants and our typically developing participants, because even people who don't have autism carry some genetic load for autism. We calculated polygenic scores using two separate approaches to assess the robustness of our results. We used both a clumping pruning approach in PRECISE and a Bayesian approach in LD-PRED, and our results were highly similar across the two approaches. In autistic boys, greater genetic load for autism was associated with stronger connectivity between the salience network and somatosensory areas, specifically the postcentral gyrus and the supramarginal gyrus. In autistic girls, however, there was no significant relationship between polygenic scores and salience network connectivity. In typically developing boys, greater genetic load for autism was associated with stronger connectivity between the salience network and lower order areas, specifically motor areas such as the precentral gyrus, which is primary motor cortex. With greater genetic load, typically developing boys also displayed weaker salience network connectivity with inferior temporal gyrus, which is part of the ventral visual stream. So as a whole, genetic load impacted sensory and motor areas in typically developing boys. Although in typically developing girls, there was no significant relationship between genetic load and salience network connectivity. So how do we interpret these findings? That as a whole, greater genetic load for autism is associated with increased connectivity between the salience network and sensory motor areas. Earlier, I showed you the social motivation theory of autism. There are also sensory motor theories of autism. These sensory motor theories posit that the emergence of autism is driven by differences in lower order sensory motor processes that affect early patterns of attention and ultimately lead to autism. For instance, if infants find sensory stimuli such as noises in the environment particularly salient, if they're particularly attention-grabbing, then maybe infants are paying attention to this non-social sensory information instead of social information. And that leads to fewer opportunities for social learning and ultimately the social differences that we see in autism. Recent work looking at infants has provided support for these theories because infants who are at elevated likelihood of developing autism, that is, infants who, based on their family history, carry greater genetic load for autism, demonstrate increased connectivity of the salience network with sensory motor areas. And our current results are very much in line with these previous findings and this theory, suggesting again that greater genetic load for autism is associated with increased salience network connectivity with these lower order areas. This is particularly notable because the current study and a previous infant study used completely separate samples and also different ways of assessing genetic load for autism, yet they yield very similar results, which are both in support of sensory motor theories of autism. Together, this suggests that if we could identify infants who display alterations in sensory motor processing, or who seem to be at a higher chance of having sensory motor differences, then maybe we could develop early interventions tailored to this population. Now that we've looked at how genetic load for autism relates to connectivity within each group, we then look to see whether the relationship between polygenic scores and connectivity differed between boys and girls. When directly assessing sex differences in the association between genetic load and functional connectivity, we found significant sex differences in both the autism and typically developing groups. Starting with the autism group, there was a significant difference between autistic girls and boys in terms of the association between genetic load and brain connectivity, specifically in terms of salient network connectivity with motor areas, such as the precentral gyrus, and regions related to sensory motor processes, such as the mid-insula. Specifically, boys with autism exhibited stronger functional connectivity between the salience network and these sensory motor regions, the more genetic variants that they had for autism. We see a similar pattern results in the typically developing group, where the impact of genetic load on salient network connectivity with motor regions is dependent on participant sex. Specifically, typically developing boys display increasing connectivity between the salience network and motor areas with more genetic load, where the typically developing girls didn't exhibit any statistically significant relationship between salience network connectivity and polygenic scores. As a whole, these findings in our autism and typically developing groups suggest that boys are more sensitive to genetic load for autism than girls. By demonstrating that the neural impact of genome-wide load is significantly modulated by sex, these findings propose an in vivo mechanism for why females may require a greater genetic load to develop autism, which has important implications for finding early screening and developing novel early interventions. And it's particularly interesting that the areas implicated in these analyses are motor areas, as both girls with autism and typically developing girls show fewer restricted and repetitive behaviors than their male counterparts on average. Together, this suggests a possible cross-scale mechanism for why males seem to be more sensitive to genetic load for autism. If we start with males, and with the fact that some individuals in the population just happen to have more common genetic variants associated with autism, you see that individuals who have more polygenic load have increased salience connectivity with motor areas, which may in turn potentially lead to repetitive motor behaviors that are common in autism. If we flip this and look at this through the lens of girls being less likely to develop autism, salience connectivity with motor areas may be less sensitive to genetic load in females, which may in turn that females are less likely to develop repetitive behaviors and therefore be less likely to be diagnosed with autism. This proposed mechanism should be tested in infants to assess whether neural sensitivity to polygenic load does indeed mediate the emergence of repetitive behaviors and the ultimate diagnosis of autism. And if so, it could have implications for development of future early interventions. In sum, for this series of studies, we demonstrated that sex, age, and genetic load significantly impact neural alterations in autism. As a whole, these studies show that understanding heterogeneity in autism not only improves our understanding of brain-based variability in autism, it also improves our understanding of autism as a whole. I'm now going to move on to our work looking at white matter microstructure in autism. Previous diffusion MRI studies in autism have generally found altered white matter microstructure compared to neurotypical controls, but the affected regions are inconsistent across studies, which may in part be due to sample sizes. Such prior work has also mostly used analytic approaches that limit our ability to accurately localize such differences within tracks. Furthermore, most diffusion studies in autism have used the traditional microstructure model DTI. However, DTI has well-established limitations regarding its inability to resolve complex fiber configurations. Here we therefore analyzed a large dataset of autistic and neurotypical participants using an advanced analytical method that allows for the precise localization of white matter differences within tracks, in combination with an advanced microstructure model that goes beyond the DTI model. So I'm now going to get more into the details of the study, which was led by my PhD student, Gowan Kim. For these analyses, we included over 350 subjects with and without autism from 10 different sites. We used standardized protocols to pre-process each participant's diffusion-weighted MRI scan before generating whole brain tractograms and extracting commissural and association tracks, of which you can see a subset here. We then applied the long tract analysis method bundle analytics, which was developed by our collaborator, Bram Shashandio. For this, we started by creating tract profiles for each bundle by mapping our microstructure metrics into all points of the bundle streamlines. We used metrics from two different microstructure models, the conventional model DTI and the advanced model TDF. TDF addresses the limitations of DTI by using a continuous mixture of tensors to account for multiple underlying fiber populations. And like DTI, it produces a measure of FA. Next, we divided each tract into 100 segments along its length to allow for the precise mapping of group differences. Our group-level analyses then investigated where the autism and neurotypical groups significantly differed along the tract, while including standard nuisance covariates and correcting for multiple comparisons. Lastly, we assessed the statistical impact of including additional sites by creating QQ plots to visualize p-values across increasing numbers of sites. I'm going to start by talking about our general pattern results for which white matter metrics were different in autism. I'll then dive more into the functions of the implicated tracts, and then the spatial specificity of our results within each individual tract. So, starting with our pattern of findings across metrics, we found that FA was lower in autism, both when using the DTI and the TDF version of FA. We also found that MD and RD were higher in autism. Together, this may suggest less efficient neuronal communication in autism on average. We found this pattern of decreased FA and increased MD and RD across all of our examined commissural tracts and almost all of our examined association tracts, where here you can see the significant tracks. And don't worry about the colors of the tracks yet. I'll be talking about that in more detail in a couple of slides. When comparing the DTI and TDF models, we found that TDF more sensitively captured group differences in many tracts, and especially so in commissural tracts. And this finding that TDF is more sensitive than DTI is also consistent with prior work looking at other scientific questions. I'm now going to dive more into the tracts that we found exhibited altered microstructure. As you can see on the prior slide, these tracts interconnected a wide range of brain regions, including frontal, temporal, parietal, and occipital regions. And overall, these tracts are known based on prior literature to relate to a wide range of functions, which I'll go through now. Starting with the commissural tracts, the mid-body of the corpus callosum is known to underpin motor and cognitive functions, whereas the forceps major is associated with visual processing, and the forceps minor has been related to executive function. The alterations observed in these tracts may therefore underpin a range of the behavioral traits commonly observed in autism, including sensory motor differences and executive function challenges. Moving on to association tracts, the unsonate fasciculus is thought to relate to socio-emotional processing as well as language, and the arcuate fasciculus is a canonical language tract. Differences in these tracts may thus be related to social communication differences seen in autism. Moving on to the last of the association tracts, the inferior longitudinal fasciculus is related to visual processing, including the associated cognitive and affective processes, and the inferior fronto-occipital fasciculus has been related to semantic processing. The middle longitudinal fasciculus relates to the processing and learning of auditory information. So together, these tracts may be related to the altered sensory and somatic processing that is common in autism. Overall, our findings suggest that altered connectivity between brain regions may contribute to a myriad of the behavioral traits common in autism, including differences in social communication, sensory processing, and executive function. So far, I've been talking big picture about which tracks we found differences in, but now I'm going to zoom in and talk about where in each of those tracks we found differences between the autism and neurotypical groups. Strikingly, we found that these alterations were definitely localized within specific segments of these tracts, rather than uniformly affecting the entire tract. Phrased alternately, the tracts that had any differences in them are distributed across the brain, but within each of these tracts, the findings are localized. Now I'm going to show you some examples of these localized within tract findings. Here's one example, which I'm going to walk you through. Up top, you can see the tract, in this case, the mid-body of the corpus callosum, where the darker orange colors reflect smaller p-values for the examined metric, in this case, TDF-FA. In the graph below, the exact p-values are plotted. Specifically, on the y-axis, we have the negative log p-values, and on the x-axis, we have each of the 100 tract segments. For completeness, we have multiple p-value thresholds plotted as lines, and all of the findings I'm presenting survive the strictest threshold. So anything above those lines is significantly different between the autism and neurotypical groups. Together, you can see that for the mid-body of the corpus callosum, the autism group displays significantly altered microstructure only in the central portion of the tract. If we look at a different tract, though, you can see that this is not a universal pattern. Here, for instance, you can see that group differences were more notable in the lateral portions of the tract. This is just 2 example tracts and 1 example metric, but we found that within tract group differences were quite localized across tracts and metrics, where here's just a few additional examples. So taken together, we have this very complex picture of white matter alterations in autism, where they're not confined to a specific type of tract or region of the brain, but they are localized within individual tracts. At the last portion of the study, I'm now going to present our result examining the impact of including additional sites. Specifically, our results that I've been showing so far have been including all 10 sites. But given known variability across sites and scanners, does including more sites actually increase our statistical sensitivity? And the answer is yes, it does. It does increase our sensitivity to microstructure alterations. Using one tract and metric as an example, here you can see the QQ plot that we created that shows the effect of including additional sites on the overall p-values. I'm not going to go through this graph in detail for the sake of time, but briefly, you can see that the observed negative log p-values are overall smaller the more sites that you add. And again, this was the case across tracts and metrics, but here's just a few additional examples. Taken together, these findings robustly demonstrate in a large sample that autism is associated with white matter microstructure alterations that are distributed across the brain, encompassing association and commissural tracts that interconnect a wide range of brain regions. But importantly, within those tracts, such alterations are localized. Those patterns of white matter alterations in autism are spatially specific across the length of the tract. Additionally, these results demonstrate the utility of large-scale samples for capturing such nuanced patterns. As a whole, this work provides rigorous insights into white matter microstructure in autism and demonstrates the importance of leveraging advanced methods in large-scale samples. We're also actively expanding our sample size and running follow-up analyses to examine sources of heterogeneity like age and sex. We'll be presenting those results at OHBM, so stay tuned. So by robustly demonstrating specific pathways that underlie autism and precisely mapping how individual differences impact these pathways, this work provides targets for future work. I'm now going to move on to discussing our work focused on brain-based sex differences across the population. The reason we're interested in typical sex differences across the population is that sex differences are really common in a wide range of brain-based conditions. This includes autism, like I talked about earlier, but also a lot of conditions that commonly co-occur with autism, like ADHD or depression. Given these known sex differences, having a really robust understanding of typical sex differences in the brain will be crucial for understanding the etiology of these different conditions and letting us subsequently develop new treatments and tailor existing ones. Many of these brain-based conditions also show significant alterations in white matter microstructure, but previous work looking at typical white matter sex differences has been limited by smaller sample sizes or by by using only a single reconstruction model, with many of these studies using only DTI. We therefore rigorously characterize sex differences in multiple samples that together span from childhood through late adulthood. I'm going to start by presenting our results looking at sex differences across development using multiple advanced diffusion models. And this is work which I've been leading together with my trainee, Sebastian Benavides, who's now moved on to doing his MD PhD. For this study, we look at neurotypical subjects from between middle childhood through emerging adulthood. These subjects had multi-cell diffusion-weighted MRI data available, which allowed us to fit additional microstructure models beyond those I presented earlier. Specifically, here we use a DTI model and two models that had never been used before to study white matter sex differences across the brain throughout all development. These models were TDF, which can be fit to single-cell data, and NODI, which needs multi-cell data. Briefly, NODI is a biophysical model that separately models restricted, hindered, and free water diffusion. It provides measures of intracellular volume fraction, which reflects a combination of neurite and cell density, a measure of orientation dispersion, which reflects the fanning of fibers, and isotropic volume fraction, which is a measure of free water. And as a starting point, we used a standardized pipeline based on TBSS to extract white matter metrics from standard ROIs in the JHU Atlas. We found girls and boys significantly differed in multiple tract regions across the brain, and that the pattern result for each metric was consistent across white matter regions. Girls displayed greater ICVF and DTIFA in the sagittal stratum, as well as greater TDFFA in a range of white matter regions that together included association, commissural, and projection tracts. We also found that TDF-derived version of FA was more sensitive to sex differences than the DTI-derived version of FA. For tracts that showed sex differences in diffusivity and free water, boys showed greater diffusivity, that is greater MD, AD, and RD, and greater ISOVF across white matter regions. In this sample, there were largely no significant age by sex interactions. The one exception was fiber dispersion in the superior corona radiata, but for the other metrics, we found that our findings did not depend on age. As a whole, these findings are beginning to reveal patterns of typical sex differences in white matter microstructure during development using advanced measures that hadn't been used before. And these results are in line with another study we completed, specifically looking at sex differences in late childhood. For this study, we leveraged data from a population-based sample of over 11,000 subjects, and we split this sample into discovery and replication cohorts to directly demonstrate the reproducibility of all findings. Their diffusion scans, which were optimized for the RSI model, were analyzed using both the conventional model DTI and the advanced model RSI. RSI is conceptually similar to NODI, but it provides separate measures that relate to cell density and neurite density, and RSI had never been used before to study typical sex differences. We assessed sex differences in white matter tracts across the brain, and we completed a number of analyses to establish the robustness of our primary findings to a range of factors. We found significant and replicable sex differences in DTI or RSI microstructure metrics in every white matter region examined. This graph shows the results for every region and metric, but I'll give you the big picture takeaways now to save time. The impact of sex on FA was regionally specific, but sex differences in other white matter metrics were remarkably consistent across the brain, replicating our previous findings. Boys typically exhibited higher MD, AD, and RD than girls on average, whereas girls generally displayed greater restricted isotropic, directional, and total diffusion, suggesting increased cell and neurite density in girls at this age. As a whole, the most substantial sex differences were observed in association and superior corticostriatal tract ROIs. These findings suggest that sex differences in late childhood are most pronounced in tracts with more protracted developmental trajectories. Importantly, these results were replicable, as demonstrated here, and they were also robust to a wide range of potentially confounding factors. So as a whole, this study provides a rigorous characterization of white matter sex differences in childhood. Expanding on this work, looking at sex differences in childhood, we've also rigorously characterized sex differences from middle to late adulthood. For this study, we capitalized on data from a population-based sample of over 15,000 participants, and we analyzed the diffusion MRI data using multiple microstructure models, including DTI and TDF, as well as the models NODI and mean apparent propagator MRI, or MAP. Briefly, MAP is a signal-based model that estimates the diffusion patterns of water molecules without a priori assumptions about the underlying tissue. We use a fractional polynomial approach to model age, which I'll discuss more on the next slide, and we simply examine age, sex, and their interaction. For the sake of this presentation, I'll be focusing on sex effects and the interaction between age and sex. Lastly, we created sex-stratified normative reference curves for each metric. Before I move on talking about what we found, I want to briefly introduce two of the statistical approaches that we used. First, I want to talk about fractional polynomials, which we use to model age. Fractional polynomials offer a more adaptive modeling approach by allowing continuous variables to be flexibly modeled in a nonlinear manner, without specifying a priori the relationship between the independent and dependent variable. This is important because imaging analyses will commonly assume that age is linear or quadratic, but that may not necessarily be the case. As a quick example, here you can see the data-driven age model for one white matter region and metric. And here you can see how different models are most appropriate for a different region and metric. We use fractional polynomials as the basis for the sex-stratified normative reference curves we created, which I'll briefly introduce here. Normative reference curves, particularly in the form of growth charts, are a standard part of medical care, and I include examples here of the CDC growth charts for boys on the left and girls on the right. Clinically, these normative charts are really important because they let doctors easily flag people who are outliers, for instance, if a child's weight drops below the 5th percentile. For this study, we therefore decided to create brain-based growth charts using quantile regression for each white matter region and metric under a sample of middle-aged to older adults, with the goal of better quantifying brain-based variability. You can see an example of charts we created, where the different curves reflect the different percentiles marked on the right, and the gray overlay reflects the kernel density. So moving on to our results, we found that sex differences in this adult sample were widespread, but the pattern of such differences was much more nuanced than in our developmental samples. For instance, in some measures of diffusivity and restriction, women exhibited greater diffusivity and restriction compared to men. But in other measures of diffusivity and restriction, the reverse pattern was observed, or the pattern was regionally specific. Thus, we found that sex differences in adulthood are very complex and depend both on the exact measure of white matter microstructure and on the examined white matter region. When examining the relative sensitivity of different microstructure models to sex differences, we found that the multi-shell model NOTI was most sensitive to such differences. So in sum, for sex differences, we found intricate and widespread patterns of sex effects, and NOTI was the most sensitive model to such effects. Now, moving on to interaction between age and sex, as well as the impact of age differs between men and women, we found that age effects significantly depend on participant sex for many aspects of white matter microstructure. But again, the overarching pattern was very complex. For some microstructural properties, women exhibited steeper age-related degradation, whereas for some, men displayed steeper decline, and for some aspects of the white matter, it was a more mixed pattern. As a whole, when examining which diffusion model was the most sensitive to the interaction between age and sex, we again found that the multi-shell model NOTI was the most sensitive. Taken together, these findings provide a thorough characterization of sex differences in white matter microstructure during adulthood. In sum, these studies using large-scale samples and advanced diffusion models robustly demonstrate typical sex differences in white matter microstructure. When using multiple distinct reconstruction models, TDF was the most sensitive to sex effects in childhood, while NOTI was the most sensitive model in adults, underscoring again the importance of using microstructure models beyond the conventional DTI model. Interestingly, together these results suggest that sex differences may change substantially across the lifespan, because the sex effects that we found in childhood were quite different from the sex effects we found in adulthood. Across two separate developmental samples using different analysis methods and models, we found that sex differences during development were consistent across white matter regions, whereas in adulthood, they were highly dependent on the specific region examined. There is, of course, a caveat that these findings in children and adults were based on samples with different acquisition protocols, but there's prior work in smaller samples with harmonized acquisitions that suggest something similar. As a whole, These findings provide a rigorous characterization of sex differences in white matter microstructure across the lifespan and will serve as an important foundation for understanding sex differences in autism and its common co-occurring conditions. Understanding typical sex differences will allow us to propose biological mechanisms and sex-specific pathways through which different brain-based conditions may arise. I'd like to thank all of my collaborators and funders, as well as, of course, the study participants. For this talk, I decided to focus on work we had completed locally, but I did also want to give a shout out to the Enigma Consortium, which we're very active in and which is always accepting new members. For anyone who's not familiar with the Enigma Consortium, I have a couple of quick slides I'm going to show. The Enigma Consortium leads the largest neuroimaging studies worldwide on a wide variety of topics. If you have neuroimaging data that is relevant for any of these working groups and you're interested in joining, Enigma is always accepting new members. There's some links at the bottom of the slide with more information. In particular, most relevant for this audience, I'd like to give a quick shout out to the Enigma Autism Working Group, which I co-chair together with Dr. Jan Boydelaar. These are some examples of some of the seminal projects from the Enigma Autism Working Group. We have a lot of ongoing and active projects too. Any labs that have neuroimaging data from autistic participants can join Enigma Autism and both get involved in ongoing projects and propose their own projects. So if you're interested in joining Enigma Autism or you have any questions about joining, feel free to reach out to me and Jan. And with that, I'm happy to take any questions on my talk.
Katherine, thank you so much. Wow, what an amazing amount of work that you've done. This is really incredible stuff. I'm curious if you could tell us a little more about some of the findings that you found with respect to the salience network, and what do we think that means in the context of differences between typically developing children and those who have autism, and differences between boys with autism and girls with autism? Can you tell us a little bit more about that?
Yeah, definitely. So I think our results are really interesting because they are in line with infant results. So we have these two separate lines of research that are supporting the importance of the salience network as a potential pathway to the development of autism. And in fact, in particular, the fact that we found these differences in connectivity with these sensory motor areas um, that support these sensory motor theories of autism. And the example I like to give is that, you can imagine, you're a baby, there's lots of things going on in the world. And if you have this increased salient network connectivity with sensory regions, for instance, maybe then these basic sensory stimuli are much more salient compared to infants that ultimately go on to be neurotypical. So for instance, let's say you're a baby and you find your clothes are very distracting. You have this increased salience of the tactile aspects of your onesie or whatever. And then you're paying more attention to that compared to a neurotypical infant, or an infant who goes on to be neurotypical. So you're paying more attention to the fabric of your onesie than you're paying attention to the voices of the people around you. And that could contribute to not paying as much attention to social information. It could contribute to maybe more difficulties acquiring language, these differences in social communication, because you're not having this implicit social learning that's happening throughout your lifespan. And yeah, our findings with this polygenic score approach suggest that this could be a possible mechanism. It would be, we need to get larger samples of infants who are at elevated likelihood of developing autism to directly test this mechanism. But I think there's great promise in that aspect. It's just hard to get large enough samples of infants.
Yeah, it definitely is. I'm curious, like, just to keep riffing on salience here, is salience kind of in this context, does this analysis, or your analyses, give us any insights into what we mean when we say salience? Salience seems to be it's almost like it's relative to something, right? It's like something has risen to a level that bears attending to, or something. Do you get a sense that there's a threshold that's different in children with ASD than typically developing controls, and for some reason the salience passes that threshold or doesn't reach some threshold? Or what do we, I guess, how are we characterizing what we mean when we say salience? And does your analysis give us any insight there?
Yeah, that's a really great question too. And I think, um, it's always tricky, of course, when we're talking about these group differences in terms of what is salient, because it's always all relative. What a baby finds salient is going to be very different from what an adult finds salient. So when we're talking about, our sensory stimuli may be more salient, our social stimuli more salient, it's always has to be, in comparison to, for instance, infants that go on to be neurotypical because, yeah, just the patterns of what salient are highly dependent on age. Um, so our findings in terms of the salience network, they don't suggest an exact threshold. It would be very interesting to try and quantify that. Can we create a biomarker that is, okay, if you're above this threshold of salience network, if as an infant, if you're above this threshold of salience network to sensory connectivity, maybe you're at elevated likelihood, and now we can, have increased screening, for instance. I think that would be a really great future direction. And we're also interested in, of course, are there different subtypes? Maybe there are some subtypes where this signal network connectivity may be more relevant and more predictive, whereas for some subtypes, maybe it's there are other driving factors that are more important in the ultimate development of autism.
You presented kind of two areas with your work in functional neuroimaging, but also the work that you've been doing with diffusion-weighted imaging. You presented kind of two sets of findings, one based on the function, the other based on the microstructural analyses and fiber tract analyses that you were doing. Do you see any linkages between the two where one might explain the other?
Yeah, that's also another really great question. I think we're very interested in doing multimodal analyses for some more logistical reasons in terms of which data sets have which modalities. We haven't gone around to those yet. It is interesting in that, for both the resting state and the diffusion, we found these sensory differences. For the diffusion, we also found some differences that are related perhaps more to the social aspects, which is interesting as well. But yeah, definitely very, very important future direction of this sort of multimodal integration between the modalities.
And your growth chart applications, I think, are really interesting and very intriguing. Um, I can't remember with that particular data set how many subjects you used. I think that was coming from some of the UK Biobank data. Am I right?
Yeah, yeah, exactly. So that is 15,000 subjects.
Yeah, only 15,000 subjects. I'm just but also curious though, if that again being based on a British sample, does that mean anything to us in this context?
Yeah, so I think, that site was a few years ago. There definitely are larger diffusion normative lifespan sample or normative reference curves from diffusion now, including some recent work that Enigma's working on for that. Um, so I think, it's always the more generalizable and representative samples are the population, the better. In particular, for the UK Biobank, as I'm sure you know, but maybe not all of the audience knows, that is middle to late adulthood. So inherently, you can't use those charts for kids or teenagers. But yeah, there are some charts that we've been working on that include the UK Biobank as well as a wider range of data sets from across the world.
Yeah, those are super important. I'm curious if you did any comparison between, because you noticed or you mentioned the fractional kind of regression kind of approach. I believe this what it was referred to as, in contrast to a more traditional approach. Did you see any di major differences between them? And I guess, how do you know that one is necessarily better than the other?
Yeah, that's another great question. So what's really cool about fractional polynomials is that if the best fit is a linear approach, that's what it will find. So you can see inherently what is the best fit. Is it linear, is it quadratic, or is it one of these more complex shapes, so to say? And we found that there were some regions of the brain where it was actually a linear relationship, but by and large, most of them were not linear or simple linear quadratic relationships. There is, of course, this was again in middle to late adulthood, so we don't know for sure if that would be the case across other portions of the lifespan, and also was dependent on the exact tract region and metric examined. But it was very interesting to see that there were many tract regions and metrics where linear quadratic was not the best fit.
In our final minute here, are there any special things that people who are interested in data science might be interested in, or you might suggest that they bone up on before they start diving into the complexities of heterogeneous autism related spectrum disorder data types?
Yeah, that's another great question. I think, being aware of, the clinical phenotype and the lived experiences of autistic individuals is always important to keep in mind when thinking about, okay, what might be the most important sources of heterogeneity to consider.
Yeah, absolutely. Well, Dr. Katherine Lawrence from the University of Southern California, thank you so much for sharing your functional imaging and your diffusion-related imaging work and what this means in the context of various genetic components, et cetera. Just fascinating work, and it's really rich. And I think you really touched on a number of different kind of neurological nuances that are really important to understand in autism. So thank you so much for sharing that. And thank you, everybody from around the planet joining us today and watching us on YouTube via recording. We really appreciate it. Thank you so much, and everyone have a wonderful weekend. Thanks again, Katherine.
Thank you for having me.